onwards to the stages of photosynthesis, we have two main stages of photosynthesis. I'm going to categorize them as forth. First of all, we have a light dependent stage. Like the name suggests, obviously the stage requires light. And then we have a secondary light independent stage. I foreshadowed a little bit when it came to this part, so we're going to be talking about that. A light dependent stage and a light independent stage. Sometimes in the past, this was actually referred to as the light reaction and the dark reaction, but you see why it was dropped, right? Light reaction and dark reaction sometimes, but do not refer to them in that manner now. Call them light dependent and light independent stages distinctively. Okay. Right, now why do I have some space in between those two things? Here's the deal. The light dependent stage, first of all, let's write down where these things are happening. So we talked about this as well. The light dependent stage occurs in the thylakoids. We're going to be looking at that in a lot more detail, so don't worry about that. The independent stage, like I talked about, is happening in the aqueous fluid, also known as the stroma. Okay. Now, the light dependent stage can actually happen in two manners, in two methods, if I may. Not just one. I'm going to write down what those two things are. You might or might not have heard of these terms before. Either way, it's fine. Don't worry about it too much. We'll be discussing in detail and you will be understanding that well. One second, let me mute my phone. It's distracting me. Okay, right. The first thing is actually known as non-cyclic photophosphorylation. It's not a hard term at all. It's just photosphorylation. Move this a little bit down. Non-cyclic photophosphorylation, and we have cyclic photophosphorylation. Do not worry about any of these terms yet. You don't have to worry about how the terms are derived and also what it means. That will be discussed in a lot more detail. We have non-cyclic photophosphorylation, which is the first method in which the light dependent stage occurs, or alternatively, there's cyclic photophosphorylation that can occur. Right. Now, when it comes to non-cyclic photophosphorylation, we usually say that, so PS is photosystem. So from this point onwards, if you see me using the abbreviation PS, that would mean photosystem. What exactly is a photosystem? Remember, it's exactly how these pigments are lined up. When these pigments are lined up in this manner, we call it a photosystem, or PS for short. Non-cyclic photophosphorylation is known for the involvement of photosystem 1. So we have two photosystems. If none of these terms make sense, that's okay, just hold on. This involves photosystem 1 and photosystem 2. We usually say that cyclic photophosphorylation only involves photosystem 1 only. Photosystem 1 only. Now, don't worry about what photosystem 1 is, what photosystem 2 is, what on earth photophosphorylation is. Don't think about any of those two things. All you have to know is these are the basic fundamentals of the stages of photosynthesis that you'll be talking about. Now, the light independent stage, the light independent reaction, is also known as the Calvin cycle. This does not require any form of light, and this is known as the Calvin cycle. Now, if I were to give you a very rough sketch of what is happening around these stages, it would look something like this. Now, why exactly do we need two different stages? So if we have a light independent stage that does not require light, is it not possible that we carry out photosynthesis completely without light? Those are probable questions, right? The answer is no, obviously. Let me tell you why. So the light dependent stage occurs first. No, we have the light dependent stage occurring. And afterwards, we have the light independent stage occurring, the Calvin cycle. Calvin cycle. Okay, here's the deal. For this cycle to go through its metabolic processes, it needs the products that are formed from the light dependent stage. That's the catch. What exactly are those products? We'll be talking about those products in detail, but as a very short, brief summary, first you need energy in the form of ATP to carry out the Calvin cycle, and you need reduced NAD. You need reduced NAD. These things can only be produced by the light dependent stage, and the light dependent stage can only occur if light, it doesn't have to be sunlight specifically, but light energy is present. That's the key idea. Now let's focus on the light dependent stage. The light dependent stage is occurring in the thylakoids. We will separately look at what non-cyclic photophosphorylation and cyclic photophosphorylation is about, what they do, why they are different, and this photosystem business as well. So first of all, let's focus on the light dependent stage and non-cyclic photophosphorylation. Cyclic we'll do later. Let me write down the subtopics. So first, light dependent stage. This is the first stage. And under this, we will talk about non-cyclic photophosphorylation first. The idea behind non-cyclic photophosphorylation is that there's no cycling of something. I won't tell you what that something is, but that's the idea. So in cyclic photophosphorylation, something is cycled within the system. You will clearly see what that something is. Okay, so non-cyclic first. The best way that I have found that I can teach this to anyone in a way that they can understand the most was always a diagram. If you follow through with this diagram and you memorize this diagram, constructing a paragraph out of this diagram to answer any form of question that you are presented under this particular subtopic is going to be light work. So let us draw the diagram first. Now, the light dependent stage, one short thing is missing, so occurring in the thylakoid. There you go. Okay. So it's occurring in the thylakoids. No? So I'm just going to draw one thylakoid, just one thylakoid. It's going to be highly detailed as well, right? Let me draw. Think that space should be enough, and let's curve back. And yep, there you go. I'm gonna try to make it a little bit more. No, okay, never mind. Okay, I don't like that. Okay, that's better. Okay, that is a thylakoid. Now I'm going to zoom in and tell you all the structures that you're going to find on the thylakoid membrane. What's the thylakoid membrane? This would be the thylakoid membrane, and this is typically referred to as the thylakoid lumen. Thylakoid lumen. Okay, right. First of all, let's clarify this photosystem business. Now, you know a photosystem is when these pigments are aligned as so. Accessory pigments and the primary pigment or the reaction center. Now it would be very weird to draw it like this, so I will be using simple rectangles to represent photosystems. Okay, so when you go from left to right on this thylakoid, on the membrane, you're going to find your first ever photosystem, and it will be located somewhere over here. Now you would imagine this is photosystem 1, but this is actually photosystem 2. They were named according to the chronological order in which they were discovered. So from the time period that they were discovered. You don't want to know any of that. That information is not required. All you have to know is on the left, starting from left, going all the way to the right, what you have is photosystem 2. Now, some small noteworthy mention that I will give about photosystem 2 would be that photosystem 2 is known as P680 as well. Now, what do photosystems do? Photosystems absorb light and transfer energy. P680 simply refers to the wavelength, meaning this photosystem is best adapted to absorb light of wavelength 680 nanometers, basically. No big deal. And then on the right hand side, so now these photosystems are going to be associated with different proteins. I will not be drawing everything and you will not be requiring all of that. I will only be drawing the things that you're going to need for your exam. And this is the second protein that you're going to need. Naming can come later, and afterwards, after that protein, we have our next photosystem. That is photosystem 1, not 2, but 1. Okay, and this is known as P700, best adapted to absorb light of wavelength 700 nanometers, so precisely red light, if you may. And then we have another little bit of proteins. And then finally, a very special and very important protein. A very special and very important protein. Let me actually rewrite these things in a better manner. I don't know why I'm lacking and slacking with my handwriting. Okay, this is photosystem 2, and this is typically referred to as P680. You've got photosystem 1 here, P700. Okay, and this also looks, oh my god. Okay, cool. Maybe not. Okay. The final protein is going to be the most important one out of all of these things. Now, if you went through cellular respiration, energy and respiration, which was chapter 12, you will not be a stranger to this protein. This protein looks a little something like this. Those of you with experience will straight away be able to recognize that this is ATP synthase or ATP ACE for short. Now let's do the labeling part. This is ATP ACE. It's ATP synthase, I'll just call it ATP ACE. These are just transporter proteins. There's no specific names associated with any of these things. This is photosystem 2. This is photosystem 1. All right. Those are the basic parts. All of these things together, we commonly refer to them as the electron transport chain. As the electron transport chain, just like in cellular respiration and the mitochondrion. So electron transport chain is a very common term. Anything that can transport an electron is typically going to be referred to as the electron transport chain. A electron transport chain. And we have electron transport chains everywhere basically. That's the thing. So now let's actually look at what's exactly happening at each of these proteins, photosystems, and what's the fate of the final ATPase that is present here. First, I will be doing a very lenient diagrammatic representation for the betterment of your understanding. I will also be getting rid of these labels for now. Electron transfer, I'll leave these two things behind like that. And let's see what exactly is happening with all of these things. So, the first thing is that light energy has to be present. Light energy has to be present. Actually, let me get rid of these labels as well. Okay, that's photosystem 2, 680. Just keep that in mind. The first thing that's going to happen is light energy. I will repeat this again. It doesn't specifically have to be sunlight. It can be light of any form of energy. It's going to come and hit this photosystem. So light energy is hitting photosystem 2 for starters. And when this happens, a small process known as photoactivation occurs. Photoactivation. The process known as photoactivation occurs. Now, photoactivation is all about light energy hits and the electron is excited. So the excitement of this electron I'll show like this. What color can we use for an electron? We'll use a bright red for an electron. It's here and it's going to get excited to a higher energy state like that. The electron is going to get excited to a higher energy state like this. This is the overall process of photoactivation, and this is the initial step of any phosphorylation, not just cyclic, not just non-cyclic. There are other forms of photophosphorylations and phosphorylations throughout our metabolic systems in our body and plants. When it comes to any of those things, this initial excitement of an electron is the term that we usually give photoactivation. Now, remember how I told you that in cyclic and non-cyclic, the difference lies between how things are recycled? I told you that something is recycled. So, no more gatekeeping. It's the recycling of this electron. If the electron is recycled, you call it cyclic photophosphorylation. If the electron is not recycled, that's non-cyclic photophosphorylation. No joke. It's as simple as that. But you are going to have to be very specific about how those things are going to be recycled. I will specifically mention the things that you have to know at given points. Now, hello. After this excitement, this electron is going to have a lot of energy. Let me drop this pen size by a little. Okay, that's better. Now, this electron has a lot of energy in it. What type of energy? Basically, kinetic energy after the electron has been excited. Now, kinetic energy is the energy of movement. No, he doesn't want to stay in the same place. He wants to move all around. But tell you what, all these membranes, they're impermeable and stuff. No. So it's the electron is going to take the only path that is provided to it. The only path that it can actually travel through, and that would be through the electron transport chain. So I'm going to illustrate its movement like so. So the electron will travel through these proteins, and all the way to photosystem 1, all the way to photosystem 1. I might as well draw that near the, so boom, boom, through this, near the edge. So here, right. Now, do you see? It's moving through all of these in this manner. There's a gradient. It's dropping. What's happening? The electron is transferring its energy to these proteins as it moves along, and therefore it loses its energy and it's back at the bottom. It doesn't have energy, that's why it has dropped to the bottom. Now, why exactly is the transfer of this energy to these proteins important? That's because this is what happens. Now, in this case, when this specific protein gets energy, this is capable of pumping protons, typically referred to as H plus ions, into the thylakoid lumen. This is a very similar scenario to what is happening in cellular respiration, where you have the proton pump. It's basically the same process. So we are pumping H plus ions, referred to as protons, into the thylakoid lumen. What is the thylakoid lumen? This inside part. That is why that transfer of energy is required. Simple summary: photoactivation, electron gets excited. I'm going to provide a proper paragraph of information afterwards of what is happening. This is the diagrammatic, schematic representation that you have to get into your head first, because this is, like I said, not a complicated process, but simply speaking, this is a very chronological process. You have to know specifically what's happening at what order. So here, the electron is excited, energy is transferred, H plus ions are pumped like so, and the electron's ultimate fate is right here. What has the electron ended up in? That is another photosystem. Photosystem 1 to be specific. What do photosystems contain? They contain accessory and primary pigments. What do those pigments do? They are capable of absorbing light. So once again, another form of photoactivation happens. Once again, light energy hits the photosystem. Same deal. The electron will now again regain the energy it lost. Excited, it moves, and boom, it comes over here. It simply comes over here and moves forward. What is this? Now, this is actually a very special protein. This protein is capable of converting something. I'm going to pick a color that I didn't use here. I'll pick an orange. Yeah, let's do like, okay, there you go. Now here, this protein is capable of converting NAD. What do you think? NAD. NADP to NADPH. Typically in cellular respiration, aerobic respiration, what we had was NAD to NADH. This is photosynthesis, no. So therefore, we just have a P. You think of it that way. You don't want to know any further information about that. NAD is converted to NADPH. NAD is converted to NADPH. Now, previously I talked about this a little bit in this stage as well. And I specifically mentioned NADH because I didn't want to introduce the NADPH to you yet, but you have to know in photosynthesis we don't talk about NADH, we don't talk about reduced NAD, it's always NADPH. I wanted to give that distinction like so. So from the light dependent stage onwards to the Calvin cycle, we're taking ATP as well as NADPH. Okay, this protein is capable of reducing NADP and forming NADPH, which is the reduced form of NADP. Reduced form of NADP. Alright, that is the ultimate fate of the electron. It goes on until the point. Now, this process is more complicated than this if you actually look at it on the quantum level and the cellular level. But this is all you have to know for your advanced levels. So I might as well stop there without giving you excess redundant unwanted information. And if your minds do pique curiosity, if you have curious minds, you can always go give a little Google search and you will get all the information that you want. Okay, now if that's the end, where's my ATP? Where's my energy? It's not produced yet because the electron, that's the only thing that the electron is capable of doing. Those are all the things that the electron is capable of doing, meaning this. Those are the only things. Electron knows how to travel through the photosystems. I will also bring back the labels here. It's looking a little bit. Photosystem 2, photosystem 1, right. Electrons are moving through, and as a subsequent result of that, all we have is this. A buildup of a protons, a buildup of a protonic gradient. Remember, we referred to this as the protonic gradient. Why? It's simply because there's a lot of protons here and nothing outside. Basically, a very less amount outside, thus creating a gradient. That is as simple as it gets. No case? Right, now what would be the ultimate fate of these H pluses, these protons? It is as before. These H pluses are going to travel through the ATP synthase. Now, you see there's a gradient, no, there's a buildup. It has to be neutralized because nature always loves balance. You don't really want to keep something unbalanced in a gradient type of form. It always tries to go for balance. It always seeks balance. Now, the problem is these H pluses cannot go back through this, cannot go through the lumen. Those are impermeable. It can only go back via this channel, the ATP ACE channel, and so it does. Now H plus is returning, and if a protonic gradient is reenacted using ATP ACE, something else is also going to occur. And that's something else. Why do I always run out of colors? Purple? Okay, any purple. And that something else is the production of ATP by the combination of ADP plus inorganic phosphate. ADP plus PI gives you a TP. And this process, if you recall, is referred to as chemiosmosis. It's referred to as chemiosmosis. The production of ATP using a protonic gradient by the combination of ADP plus inorganic phosphate. This combination, so the combining of these two things, they require energy. That energy, these are all the systems that are in place to provide that energy so that ATP can be resynthesized. Now let's just look at what we actually produced by this. So we have now ATP. What else? We produced NADPH. Do not mix this up with NADH, okay? NADH is for cellular respiration. Here we're talking about photosynthesis, so you just remember the P, that's all. And NADPH. Going back to this, what did we need from the light dependent stage? We wanted these two things. Now, do we have those two things? Yeah, we do. So is that the entire process? That is 90% of the process right then and there. It's not a complicated process if you learn it in this particular manner. And that is the overall process of non-cyclic photophosphorylation. I have emitted a small thing out. I'll talk about that a little bit now. Now, do you see if this keeps happening? So if photoactivation keeps happening and the electron is excited and it travels and re-excites and it just ends up here and into NADPH, we're going to have a problem, because if more and more and more and more photoactivation occurs, this photosystem will lose so many electrons. Do you see? This electron doesn't come back to this. It just vanishes. It just goes away. It completes its process. So then photosystem 2 is just going to keep losing electrons. We don't want that. That's not a good thing. So photosystem 2 has actually adapted a specific method. Now, I will elaborate on that here. The thing is, in photosystem 2, we have an enzyme. We have an enzyme known as the OEC. The OEC is typically referred to as Wolfing complex. You can call this the OEC for short, no problem. Now what this does is, this enzyme is capable of splitting water. It splits water to replenish the lost electron. So it's going to now be able to replenish this excited electron. The process looks something like this. We take water and it's going to split it into oxygen. That's why this is also termed the oxygen evolving complex, because the ultimate result of this enzyme results in oxygen, the production of oxygen. H2O, O2, and the hydrogen is split like this into 4 protons and 4 electrons. Into 4 protons and 4 electrons. Okay, this process is known as photolysis, breakdown of what? That was photoactivation over here. This is known as photolysis, where we split the water in this manner. You might as well just split this into O2 plus hydrogen. Yes, but that's not what this enzyme does. It wants to produce electrons. No? So as a result, we're going to have some extra H pluses as well. No issue. H pluses are fine. Okay, H pluses are fine. Now if you look at this, it's not balanced. So to balance this, I'm going to do that. 2H2O, O2, 4H plus, known as photolysis. Okay, photolysis. That is how this photosystem will not run out of electrons. Otherwise, if this keeps happening without the involvement of this enzyme, this is going to happen. This not happen. This is going to run out of electrons and the process would stop. So error 404. Biological systematic error, I guess. Something like that. If you are really excited, I mean, when it comes to a research basis, you could possibly conduct a research to see what happens if you remove this specific enzyme from the system and try to run photophosphorylation, whether it works, whether it stops, whether some other things come in the way. Always a possibility. Okay, let me talk about this conversion a little bit. So we've got NADP converting to NADPH, no? Like so. This conversion, I'll talk about that a little bit. Now, H plus ions, so protons and electrons from photosystem 1 combine with NADP to give NADPH. What does that mean? So we've got these H plus ions, some extra H plus ions, this electron, they just combine to give NADPH. So I will also write down the equation for that over here. I'm going to increase the thickness of the brush a little bit. NADP combining with 2 hydrogens and 2 electrons to give you NADPH. NADPH. Those are the overall reactions that are taking place in the thylakoid membrane and the thylakoid lumen to bring about non-cyclic photophosphorylation. And hence this is a detailed breakdown of what is happening in a paragraphic form. First of all, light energy hits the accessory pigments. We know that's how it happens. And then that energy is going to be transferred onto the primary pigment, which is chlorophyll A. Photosystem 2 is also known as P680. Why? Because it's mostly adapted to absorbing light of wavelength 680 nanometers. Electrons are excited. We call that photoactivation. Always when referring to this, always think of the diagram. Do not memorize paragraphs. That's not going to help you. This will lay out what's happening. That's going to be very important in a proper manner, but always refer to the diagram. Photoactivation, electron excited, electron moves. So following through, you will see accepted by the proteins in the electron transport chain. It travels through the electron transport chain all the way to photosystem 1. As this moves, the electrons provide their energy to these proteins, and that energy is required to pump protons from the stroma, the thylakoid lumen. By doing all of this, now this electron has depleted its energy. Now remember, it comes back down to this level. It needs to be re-excited. How exactly does that happen? Re-excitement happens at photosystem 1, again by photoactivation. It's a photosystem. It contains photosynthetic pigments. It's completely capable of absorbing light. And then after all of that done, the electron comes to its end, the end of its pathway. This electron along with H plus ions are used to reduce NADP all the way to NADPH. And now by doing all of that, remember, we have a H plus or a protonic gradient, an imbalance produced. It's a huge gradient. And H plus ions move back into the stroma. Why is it streamer? My God. 1 second. It's not streamer, it's stroma. There you go. Stroma via ATP synthase or ATPase, and the energy given is thus used to synthesize ATP from ADP. Whenever we synthesize ATP by combining ADP with inorganic phosphates, we call that process chemiosmosis. This is also occurring in the mitochondrion. Oxygen evolving complex, remember, it prevents photosystem 2 from losing so many electrons, replenishes those electrons. If too many electrons are lost from photosystem 2 will get oxidized, because remember, oil rig, oxidation is loss of electrons. If too many electrons are lost, photosystem 2 will be oxidized. That's a problem. We don't want that happening. Photolysis of water is what we call it. And then what are the final products? After all of this is done, what we get is ATP and NADPH. We can, without any issues, feed this into the Calvin cycle. So we have two products going, ATP and reduced NADP, which is NADPH. That's on the Calvin cycle. And look at this last statement. Observe this. Light hits photosystem 2. Okay, it's involved. It moves, and we need re-excitement from photosystem 1. Once again, there's photoactivation there. Because of that, you can clearly see that both of these photosystems are involved. Make sure when it comes to non-cyclic photophosphorylation, your final statement of your answer will always be that photosystem 1 and photosystem 2, they are both involved. That's very important. You'll see why when I talk about cyclic photophosphorylation, but here you have to talk about this as both photosystems being involved. Both photosystems being involved. That is very important. So that was non-cyclic photophosphorylation. Let's see what's happening in cyclic photophosphorylation. Now, tell you what, you don't have to remember a completely different diagram for cyclic photophosphorylation. It's going to be the exact same diagram. as non-cyclic photophosphorylation. So let me just do a little bit of magic. It's going to be the same exact diagram. There's no difference. I mean, the process is slightly different. I'll tell you why and how, but not many things are different. First of all, cyclic photophosphorylation talks about a cycle. And like I said, this is the cycling of an electron. Look at the process and look at what is happening. This stage is as is, no difference. Photoactivation occurs, light energy hits photosystem 2. The electron gets excited. I'm going to zoom in. Electron gets excited. It moves like this all the way here. H plus ions are pumped through the lumen all the way onto here, and then it moves, re-excites itself, and moves like this. This is where the process differs. Now, if it was non-cyclic photophosphorylation, this would go here and to NADPH. That would be its final fate. But this final movement does not happen. So I'm going to get rid of that, and I'll get rid of our electron. No, not that. Okay, there you go. No. And here what's going to happen is once this electron comes here, this electron will be recycled. This electron will be recycled not to photosystem 2, but think of it as being recycled just to the electron transport chain. Just to the electron transport chain. Okay, it will be recycled to the electron transport chain. Let me make a small amendment. I will redraw the re-excitement and then draw it. Okay. Recycled to this. So now do you see, it doesn't go this way. That does not happen. There's no this form of movement anymore. So because of that, this guy cannot get reduced. We will not be having NADPH being produced. So that means NADPH is not produced. Only ATP is going to the Calvin cycle in cyclic photophosphorylation. That's important. All of the rest of these things will be happening. H plus moving, chemiosmosis, ATP will be produced. Only that produced and traveled ATP will go on to the Calvin cycle. There will be no NADPH produced. Now, there's actually many arguments about whether this photosystem excites it and then moves it, whether it does not excite it and move it, whether this actually travels to the electron transport chain or to the photosystem itself. There's a little bit of a scientific dilemma there actually. What I'm going to tell you is you don't have to focus on that. For your level, all this information is more than enough. You don't have to be focused on the very specific mechanics. Science is always changing whenever we find new things, so don't worry about that too much. All you have to know is here, it starts off like this, and the difference lies here. The difference lies in this loop. So simply you can call it that the electron is recycled, though I'll just call it electron is cycled. Electron is cycled like so. Noteworthy mentions, we have no NADPH, nothing. And now do you see here, this photosystem excites one electron and moves it like so, and it gets cycled over and over and over and over again. Is he going to constantly keep losing electrons? No, right? There's no risk like that. So because of that, this step is also not required. Now here we are losing electrons, no? So we had to do photolysis to replenish those electrons right here. But when it comes to this, there's no requirement like that. So you do not have to have a photolysis step. So I'll call it that. You don't have to have any form of splitting or photolysis of what? Not required. Only ATP is traveling to the Calvin cycle again. Okay. Only energy is produced. Keep that in mind. Only energy is produced. There's no NADPH being produced. So that is the overall process. You don't have to remember two diagrams. Remember the same diagram and the difference in the processes and where it differs. So I'm also going to do something like this. Let me bring this text all the way over here. Let's align it as so. So I'm going to go through this and tell you what's different. So this happens, okay? Light energy hits photosystem 2 and primary pigment reaction center. All this happens as is. There's no difference here. Photosystem 2 is involved in the beginning. And there's a catch. I'll talk about that later. Electron is excited to a higher energy state. That would be photoactivation. Proteins, electron transport chain. Electron transport chain proteins are going to gain energy. Pump H ions from the stroma to the thylakoid lumen. Okay. This step also happens by photoactivation. This electron will replenish its energy. Now here, this step will not happen. There is no NADPH produced. Instead, the electron is now recycled all the way back to the electron transport chain. That is one of the defining features of cyclic photophosphorylation. Okay, this thing also happens. Yes, chemiosmosis. Does this happen? There's no requirement for that to happen, so we'll get rid of that. Are both of these things produced? I don't think so. Only ATP would be produced. There's no involvement of NADPH. Only ATP is passed on to the Calvin cycle in this case. Here's the deal. Here's the most confusing part. Now, if you have a brain and you can put 2 and 2 together, you can see obviously we're starting off with photosystem 2 and then photosystem 1 and then the cycle. So this final statement is completely valid. Final statement is completely valid. Photosystem 2 and photosystem 1 are both involved. But there's this debate over here. And from analyzing years upon years of mark schemes, the thing is, when it comes to this, we don't really talk about this that much. Sometimes this process can happen in photosystem 1 solely. Sometimes, but this is the structure of how it happens. Don't have any doubt about that. This is how it occurs. But that's the thing. Sometimes this involvement is not talked about at all. So therefore, what we usually say is photosystem 1 is only involved. Photosystem 1 is only involved. Only photosystem 1. Okay, let me rephrase that. I'm losing my grammar now. Only photosystem 1 is involved. Only photosystem 1 is involved. So I'm going to highlight the special things when it comes to cyclic photophosphorylation in this color. The recycling part and the involvement of photosystem 1 only, and only ATP being passed on to the Calvin cycle. Now here photosystem 1 and 2 are both involved. There's no doubt. You can clearly see that it's going to constantly keep occurring and occurring like this. No difference. Here, because of this recycling thing only, this photosystem 1 involvement is only present. Now my advice to you would be, don't think about it too much. Don't try to be too literal. Just remember that photosystem 1 is only involved. You have to state in a mark scheme, in a question paper, in an answer scheme, that only photosystem 1 is involved. There is no involvement of photosystems 2, while you can clearly see there is the involvement of photosystem 2. Like I said, sometimes this entire process can happen in photosystem 1 only. So you can kind of think of it that way if that helps, but don't try to think of it too much. That would be my advice. I remember once losing my mind over this, trying to figure things out. So what I usually tell my students and give you advice would be to not focus on that too much. Non-cyclic photophosphorylation, both photosystems are involved. That's fine. Cyclic photophosphorylation, only photosystem 1 is involved, and that would be the final point for the light dependent stage of photosynthesis.